The human colon houses over 100 trillion microbes, more than 10 times the number of human cells in your entire body. We know these microbes have a substantial impact on our health, and nowhere is this more clearly demonstrated than when these microbes are disrupted, such as in Clostridium difficile infection, or CDI. In CDI, the bacterium C. difficile flourishes in the colon following antibiotic treatment, leading to abdominal pain, diarrhea, or worse, in more than 500,000 individuals in the US each year. Unfortunately, standard therapy for CDI, more antibiotics, fails to clear the infection in nearly a quarter of cases. Well, fortunately for these patients, fecal microbiota transplantation is emerging as a highly effective therapy for CDI. In FMT, or fecal microbiota transplantation, fecal material from a healthy donor is delivered into the colon of a patient with CDI, leading, in over 90% of cases, to a complete cessation of symptoms and no further recurrence of the disease. However, despite the efficacy of FMT, we don't understand how it works, which limits both our ability to design even better treatments for this disease and limits our understanding of the role our colonic microbes play in our health. My thesis aims to investigate the mechanisms behind FMT and therefore bridge the gap in our knowledge. I began by investigating the types of shifts in fecal metabolites after the procedure, which might indicate shifts in the functions of fecal bacteria. I found that the most dramatic changes were in the types of bile acids present in the feces. Now, primary bile acids are produced by the liver, and their main function is to aid lipid digestion in the small intestine. When these primary bile acids reach the colon, bacteria transform them into secondary bile acids. My study showed that there was a significant decrease in primary bile acids in the feces after FMT and a significant increase in secondary bile acids. Now, importantly, previous work had shown that some of the primary bile acids can cause C. difficile to germinate, to undergo the process of transforming from inert spores into active vegetative cells that cause disease. In contrast, some of the secondary bile acids prevent the same process. Therefore, my work may have indicated that a shift in fecal bile acid composition may be a mechanism explaining the success of FMT. Moving forward, I'm investigating, testing this mechanism in vitro, and also investigating whether changes in colonic bile acid composition may be a way to treat C. difficile infection. Overall, my work will demonstrate potential new therapies for this important disease, and will also shed light on the role our colonic microbes play in our health. Thank you.